capacity of Associate Professor and Head, Department of Pharmaceutical Chemistry, along with teaching and research. Dr. Mushtaque Shaikh sir also been holding important post of the NAC coordinator, and I would like to tell under his that particular leadership, our college has been accredited, completed this NAC accreditation, and we have got the A+ grade in the first cycle. Mushtaque Shaikh sir is also the president of INC Viscop. Dr. Mushtaque sir was graduated from Bombay College of Pharmacy for doctor's degree and was associated there for almost a decade with post of assistant professor. The field of research for sir are 3D QSAR, quantum chemical calculations, homology modeling, docking, structure-based drug design, synthesis, chemistry of active molecule, prodrugs and impurities are some of the key research areas of the Mushtaque Shaikh sir. In last 2 decades of his research, he has published 23 research papers, 5 review articles, 1 book chapter, 3 book reviews, and presented around 25 posters in different number of conferences and seminars. He has guided almost 19 MPharm students and is guiding also currently few MPharm and PhD students. Mushtaque Shaikh sir has received the grant around 23 lakh from the government agencies like GPU grant and from few other granting agencies. He has also received the Best Emerging Scientist in Drug Discovery Award as a best research paper for the first National Research Convention, that is Avishkar 2008. So, with this brief introduction of our HOD of Pharmaceutical Chemistry Department, I request HOD QA Dr. Anita A. Madam to please come on the stage and felicitate Dr. Mushtaque Shaikh sir by giving the certificate.
Thank you, madam, for inviting us.
Thank you so much, madam, and thank you, sir. The another resource person for this session who is also helping to Dr. Mushtaque Shaikh sir is Dr. Anand Chintakrindi sir. Sir is currently working as Assistant Professor in Pharmaceutical Chemistry, Department at Vivekanand Education Society's College of Pharmacy, and completed his B.Pharm in 2007 and M.Pharm 2011 from Bombay College of Pharmacy, and pursued his PhD in Pharmaceutical Chemistry from K.M. Kudanani College of Pharmacy. He is recipient of JRD and Ratan Tata Scholarship during his B.Pharm, AICTE Scholarship for securing AR. He is also having the AR 319 in GATE 2007 and ICMR JRF and SRF during his doctoral research. He has 10 years of experience in industry and academics across different industries like Rubicon Research Private Limited, McLeod's Pharmaceutical Limited. He has also worked with Principal K.M. Kudanani College of Pharmacy, Bombay College of Pharmacy. His research expertise include computer-aided molecular design and small molecular synthesis. He has also been hands-on experience in different biological evaluation of the organic molecules using animal tissue culture technique such as cytotoxicity studies, antiviral testing, along with the enzyme inhibition, enzyme kinetic studies at Hopkins Institute. He has presented his research work at various national conferences, including representing the University of Mumbai at Inter-University Research Festival Avishkar 2010. He has published 10 research and 1 review article in the various international high impact factor journal, including biogenetics, medical chemistry, etc. He has received academic license for fee waiver of USD $500 for Amber 18 suit from the University of San Francisco, and was granted a 6-month license for BIOVIA Discovery Studio, etc. So, sir is master in different softwares and computer-aided drug delivery and drug designing. So, with this brief introduction of Sir Anand sir, I request again Dr. Anita Ari madam to please come on the stage and felicitate Dr. Anand sir by giving the sacrament. Thank you so much, madam, and thank you, Mushtaque sir and Anand sir. I know you have both are expertise in this field, and maybe some of you are expertise in drug discovery and all that. I request you both now to start with the workshop on a very important computer-aided drug delivery system.
So, good evening everybody. So, I just start my session with a small story that was my first student Sachin Kokne was presenting his research in front of Dept. Dept was Bothra sir from Malaysia. He asked Mushtaque, what you presented, what your student presented is medicinal chemistry. Where is pharmaceutical chemistry? I said, sir, I mean, student has registered under pharmaceutical chemistry and he has synthesized some drugs and candidates. So it is pharmaceutical chemistry. He said, no, it is pharmaceutical chemistry. We should also talk about XP and taking some formulation. This kind of thing should be there in your research. So that was the time. I mean, I was starting. We are still doing that kind of science. Another story is of Professor Coutinho talking with Professor Akaman. Akaman is saying, Coutinho, stop doing drug discovery. Now it is age of drug delivery. So this is scenario is changing, and we also should change our focus as Department of Pharmaceutical Chemistry. We are trying to do that. We are doing something which are beyond let us say kind of multidisciplinary in nature. So starting with this workshop on computer drug delivery system. So let us begin with this note that many people are kind of operate or not willing to get into this field. I say just dive. People have many excuses like complex chemistry is difficult, it requires high-tech computers, requires high-cost software, and some people believe that are they really reliable? The results are reliable or not? So my take on this will be that this kind of studies they will add some rational. Seeing something will make you believe because when you do simulation on computer, you see molecules roaming around. Okay, I don't know. I was attending Professor Nagasendra Madam's talk today. I mean yesterday only she had talk, right? So I don't know if she has shared her liposome CAD or liposome. I don't know. Has she presented? Right? So they could see, I mean, liposome particles moving and how the drug candidate was indoors or kept inside that liposome. Then it adds impact. Okay, you will get higher chances of getting article accepted. It adds understanding. So today my case study is highlighting that particular point. It added to understanding. And there are many free softwares and which will work on your regular computer or laptop. So that is also not a big problem. And even eating was difficult when we were learning to eat, right? When we are young, correct? So unless until you dive in, then you are to go to learn. That is great. There are many methods. So just while ago when I came down, question answer session was going on. So I learned that lot of statistics and QBD, design of experiment, all this sort of experiments they will be done on computer. So let's say that will be one of the part of CAD's method. Along with that, you want to see how your molecules are interacting when they are in formulation stage. So we know a lot of conjugates product we make. Okay, we want to know whether they are getting hydrolyzed or they are reaching to the target site, how they are interacting with the target site. All these things can be done using blocking or pharmacophore mapping, particularly if our drug is passing through some channel or not. That's part of admit. Then QSR, so I have shown some kind of graph over there. So classification and QSR, there is another very vast field which is used in industries. Then interaction of molecules, I was talking about this at the side, and you can see the movement of molecules when they come across that barrier. And of course, many different levels of admit. I mean, all levels of admit can be studied in CAD. With this introduction, I'll now start with my case study. I was saying that this case study was focused around increasing the understanding of a particular formulation. The formulation was a cell non-emulsifying granule. The candidate was meloxicam. So what was observed that my peers, Dr. Desai at BCB, she was making one emulsion and she was trying to dissolve this drug in a very well-known lipid excipient called as labrapil M. So it was not getting dissolved, and then they read and figured it out that if you add sterilamide, little bit of sterilamide, it dissolves, and they were able to make the emulsion. So they really wanted to know, okay, what is happening, why this thing dissolves when you add a little bit of sterilamide, and they want to see that how it will, what will be the matrix we wanted to see. So objective was to evaluate the effect of salt formation on solubility of amylase in lab in presence of assay. Now where this salt will come, okay? Yeah. So the salt, when we thought about this particular thing, we thought there could be two scenarios. Either your sterilamine is interacting with that labrapil M and altering its property. That could be one case, or the case would be that assay sterilamine is interacting with MLX, the drug, okay, and enhancing its solubility, right? So the second thing looks like more logical. The second case, where the salt formation between phenolic oxide of meloxicam and amino group of sterilamine was, kind of envisaged. So we wanted to prove that whether it is happening or not, right? So that was our ultimate goal, right? So to do that, okay, we wanted to make formulation in computer. Let us see what will happen if this thing will come into a real, picture, right? So there are various ways of solving out things, right? So there is explicit and implicit solution. Implicit solution means we just consider the that solvent is present. For example, setting the properties of polarity, okay, and epsilon value of the system, it will behave like the substance was dissolved in water. There will be no water. Explicit solution means we'll put the molecules of both the entities, right? So point was there, in this case, implicit solution could not evaluate because we don't know the physicochemical properties of the solvent over here, labrapil FDP, right? And secondly, if you want to study the explicit solution at computer, there is some information you will need to feed into the computer, which we call parameters for each atom, each molecule in the given group. So that was the challenge. That was not ever reported. So first time we did it in 2017, right? So what we did, that we decided we make a small box of it. Let us say 75 by 75 by 75 and stop this small box, right, of our system, and we'll see whether how the three rings are interacting with each other: meloxicam, SLA, sterilamine, and labrapil. So we, based on their density and molecular weight, we calculated that in a box of 75 by 75 by 75 angstrom, how many molecules will fit of each one, okay, based on their concentration, which the practical scientists were doing on their bench. So this information came from them, that what concentration they have been using, okay. So there is a software called as Molecule V1. First thing we calculated that in this box, if this molecule is put, so what we have to do, we have to download the 3D structures of each one of them, okay, and submit to this software. 3D structure means all XYZ coordinates will be there. It will calculate that how many molecules of each one will fit if a particular concentration if you want to set. So all this information it will directly given: concentration, molecular weight, and the 3D structure. So it will calculate. So we know now that let us say somewhere in the somewhere is anyway. So we came to know that we'll have some 600 molecules of levrafil M. Somewhere I missed out in slides. Anyways, so we actually calculated some 600 molecules of levrafil M, some 70 to 80 molecules of stearinamine, and we decided that it will be only one molecule of eloxygen. So that ratio will do. Okay. So first thing that you want to make that box, right? So that is a software called as PackMold. It is actually a kind of add-on to a very well-known software called as VMD, which is freely available. And that is the kind of software. I mean, I'll say that is soul of this entire talk or entire work. I'll say VMD. With VMD, you can do n number of things as far as CADs are relevant. You can actually see how molecules will fit in a nanoparticle or nanotube. Okay, all this sort of experiment can be done, really done, and lot of fan base is there, so they keep on adding scripts there, right? So it is not that things are not available. They are available on worldwide, but you can download, you can slightly edit it. It is a bit of coding you will require, okay, to run and make the model. So this PackMold was available. This information it needed that how many molecules you need to put in there, right? So it created that box of 75 by 75 strong. So we got this box. You can see over here. This is VMD visualization tool. You can see that these molecules in ball and stick model, they are SLA, but the line model that is for the geographic end. So this was first system we made, and then later on we put drug into that. Okay. Now this drug. Okay, what we are saying that it will form a salt, right? So we all know that one has to be one part of the salt will be positive charge, sodium, for example, sodium plus, and the part will be chloride minus, right? So over here we are envisaging that sterile amine that will form a positive thing, okay, while oxygen will form a negative thing, right? It is hardly seen any time in literature that salt formation between phenol and amine. Okay, but that is our basis of this particular understanding. We will have to see whether it exists or not. Okay, so while teaching, such this kind of example will come in front of us. Kunal will agree when we teach non-aqueous titration. We talk about titration of amines. If you want to do titration of amine using a very strong acid, then sometimes we want to add picric acid so that it ionizes out, and the solvent will be benzene. So amine ionizing out in benzene in presence of picric acid. So that case was in my mind. So we took that leap that phenol can or even amines can be ionized in presence of some other entity. So that idea from the classroom I took into the laboratory. So here we wanted to prove that salt will form. So this is how CAD works. That you will prove how it will not work, right? So we did that thing only. We just made a mixture of two things, putting these two molecules in non-ionized state, and in another system we made the salt, means one molecule. Okay, let us consider now salt as a whole molecule, right? And then put that in the center of this particular cube, right, which is solvent over here, right, in presence of our experiment stereo. And then we did something called as molecular dynamics, right? So maybe when you enter in your kitchen someday and you figure out while cooking tea, you figure out that your sugar has exhausted, you pick up a new packet of sugar and put in that, and then at the end you figure out that little bit of sugar is still there in the packet, and the bowl has completely filled. So what we do, we shake it a little bit, so that there is space. Exactly that is the case over here. We will shake this entire system so that things will settle down. So I will just, I wanted to show 3D movement of this, but unfortunately I am not able to do that. So what you will observe that this stearinamine that has gathered in some places, that will not happen in solubilization. What we say solubilization means diffusion of drug into the voids. So it should be equally divided. So that's what we did, we shaped it, okay, for a good enough time. Let us say the time will be some 50 nanoseconds. Okay, that is a huge time as far as calculation is concerned. But real life in 50 nanoseconds is a very small time. Okay, so after that it distributed very well. Okay, so that experiment we did for both the system with MXN, I mean Loxicam plus stearinamine in non-ionized state, and one other case where they were present as salt. And then at the end we calculated the free energy. Okay, so this is our protocol. Let us say build and optimize individually the molecules, make them stable form, achieve their stable form by using minimization, create the complex as order box, do the MD calculation on that, and then calculate energy and interpret the results. Okay, so these are the results what I wanted to show you all. So what we observed, of course, it is a salt, so it was bound to be there, but we wanted to know whether solvation, because everything is hydrophobic there, if that puts or let's say overcome this ionic interaction or not. So we figured out that when it was unsalted, okay, both the things entities they ran away from each other. While other one where we kept it ionized, they were in the same place, they did not run away, indicating that some kind of ionic interaction, let us say salt bridge type is getting formed over there, right? So that was our interpretation, and in the meantime we also calculated the delta G. So there is a typical way of doing that, which we call Lambert's method. So many of you must have heard about Boltzmann equation. So same kind of equation is there for Lambert's method, where we identify the energy of each frame. Okay, now this each frame is what? Let's say I said just now that 50 nanosecond that simulation I am doing. Okay, so let us understand what is the vibration frequency. Vibration frequency, I mean, in what time that vibration of a bond electronics is 1 frame per second. Okay, so force is equal to mass with acceleration. What is it? Newton's law. Okay, so we'll apply this much force. इसमें 1 frame per second का movement हो, 1 bond to vibrate. Okay, so we provide so much force that it should move for 50 nanoseconds. Exactly, that's what we do. Okay, so it will keep on moving, right? So we'll take frame after certain iteration. So let's say thousands of iterations, thousands of pictures will be there in front of you, right? So from there, I'll choose out confirmations. So I'll get an ensemble confirmation when I do diagrams. Okay, that is the output, right? So from each frame of that, okay. So let us say, just for example, I'll put another example. When drug is reacting at a receptor, it is slowly coming. It reacts and then gets detached. Okay, so this will all frame. Sometimes you want to figure out. Okay, so coming towards and going away, this information can be used to calculate the delta G of binding and delta G of unbinding. Okay, so that is a very important information. So the same thing we did. Okay, we calculated that how stable the two systems are there. And our calculation is suggested that the right hand side system was much more stable, even energetically. Usually also, I mean, that was in hand with the information. So that tells us that there was definitely kind of salt formation occurring over there. So that is what I mean. That age we added to the paper, and it was very well accepted. It was even gotten many citations, and just in a single review, it was published. So with this, I'll stop my talk, and I am sure that I'm able to say this particular point that CADs really help you, okay, to put on your language in a proper way and make the understanding in a better way. I'll request sir to please take over guys. Thank you very much.
Okay, so basically again this is the last session and which is very challenging as a speaker to engage the audience. So I'll try to quickly wind up rather. Sir has talked about different methods which can be used. Basically when we say CADS, what is CADS? CADS is Computer Aided Drug Delivery System. Here we are applying what we are learning in CAD, that is the CAD techniques, computational techniques which are used in drug discovery. The application is there in the CADS. So there is nothing new as such, new method which we are using for CADS, but basically enhanced application or the newer application which we are using in a pharmaceutical chemistry domain or as suggested medicinal chemistry domain to the pharmaceutical chemistry domain we are looking. So I will not go in as advanced technique as sir explained. That is molecular dynamics like a PhD of CAD. I try to start with the UG level, that is the talking technique, which I suppose everybody is aware of or must have done or must have seen earlier. So basically this, as I said, this is just an extension of what we do in the chemistry domain, medicinal chemistry domain, to the pharmaceutical domain. So one basic application or very trivial application of such technique or use of molecular docking technique is to find out what we have displayed on the screen is the beta cyclodextrin complexes. Okay. So just for the sake of the starting the proceedings, we all know that what is the application of beta cyclodextrin. It is a polymer, a cyclic polymer of glucose units. Depending on the number of the units, it is classified as alpha, beta, gamma. And the central core is an hydrophobic portion which encloses the hydrophobic drug, and the outer part of that cyclic polymer is a hydrophilic portion. And with this kind of geometry of that or the topology of that particular cyclodextrin molecule, it is entrapping the hydrophobic core and exposing hydrophilic residues to the hydrophilic environment. This is basically what is happening. Okay, so quickly we will see what are the applications. Sorry, if the slide is not visible. Basically, it is aqueous solubility. You can enhance the resolution rate, you can enhance the absorption, you can enhance the permeability, and a culmination of this effects is the overall improvement in the oral viability. Okay, so what we can do, the application of molecular docking, whether first the problem statement, you will define the problem statement, what we can do using this technique. First, we can find out, is the molecule very large enough to be fitting into this particular cavity? That is the first question we can answer definitely using this technique. And if it is docking, what is the ratio of that complex? 1:2, 2:1. When I am saying 1:2, that means 2 molecules of drug and 1 molecule of cyclodextrin, or rather 1 molecule of the drug and 2 molecules of cyclodextrin. What is the possibility? Which ratio we can take? Okay. So what I've done is, since we cannot actually perform the experiment here, this particular docking experiment, I'll just run through screenshots which of the study which I have performed at docking experiment. So what are the requirements? So which softwares we'll be using? Is Discovery Studio Visualizer. This can be any visualizer. Okay, any visualizer. You can be using Schrodinger, you can be using PyMol. This is just to visualize and edit molecules. Then there is one software called as Avocado. Again, this is a minimization tool which I have used. Again, this can be any minimization. You can do the same thing in the other softwares as well. And the main application, that is the docking tool, Autodock tools and Autodock. Why Autodock? Because it is freely available. We can definitely. But there is a catch here. Right from its installation to the usage, you will see many errors during its usage. Okay, so during the course of this particular presentation, I will tell you some nuances which I as a researcher had and how I would overcome that particular part. Okay, so just now for any docking, what are the 2 components required? When you say dock, what we are docking, we are actually estimating how a particular ligand is binding to its target. Now in this case, the ligand is your drug molecule and the target here is beta cyclodextrin. Okay, so we need to understand whether a particular drug will go into that part. So for now on the discussion, the receptor for our study would be the beta cyclodextrin and the ligand is our drug molecule. Okay, so the materials required is beta cyclodextrin. Now this can be easily downloaded from the crystal structure database, that is RCSD. Okay, there are various structures out there, are just randomly picked up. There is no as such bias as such. Whichever first came into my mind, I do. Although when you are selecting structures, there are certain criteria to be looked at. Just for the purpose of study, I have not, and if I were to include that discussion itself, it would be taking hours to complete. But there are certain parameters which we look when we are selecting the crystal structure from a particular database. Basically, the first thing is the resolution of the structure. Okay, lower this resolution, that means numerically lower, better is the structure. So, beta cyclodextrin is the receptor and ligand. I have chosen ibuprofen. The PDB ID is this. Sorry again for that technical error of the slide. That what is the methodology? Any docking experiment, it is basically divided into 4 steps. That is preparation of the receptor, preparation of the ligand, the actual docking, and then the docking analysis. So I go step by step. The preparation of ligand. So basically, we start. Where is our starting point for any docking? Is downloading the receptor structure and the ligand structure. So you go to this particular site www.rcsb.com. You type in the search box the keyword which you want. Say cyclodextrin I want. So I will type in cyclodextrin. In this case, ibuprofen. I will type in ibuprofen. As I said, there are certain criteria to be looking out when you are selecting the crystal structure that you have to okay. So the box which I have highlighted, that is the download file, that you have to understand or rather click. So it will move on to this particular screen. That is you have to. There are various formats available. In the PDB format, you have to download. So once you download this, you have to save it in your computer and open this molecule in a visualizer. As I said, you can use any visualizer. I have used Discovery Studio visualizer. Okay. Now what we want, this is actually a complex ibuprofen with some particular target. We don't want the entire target, we want only the drug molecule. So I have just selected the drug molecule. Okay. Now to invert this selection, or rather to just select this, what I have done is you have to right click on that particular ligand. Okay, if you are this particular yellow part which is being highlighted, that is your ligand, and you have to right click on this, and there are several options. You have to click on the invert selection. Invert selection. So what that will do is it will deselect your molecule and it will select all the other rest of the complex. And you have to just press delete so that you will get only your ligand molecule. Now, mind you, this ligand molecule is a crystal structure. That means it has certain crystal packing forces which will distort the overall geometry of the molecule. So, certain torsion angles, certain bond angles may be distorted because of the basically the crystal packing forces. So, to relieve this pain, and before that, you should understand this is a computer which has certain lines being drawn. Okay, the computer will not understand the line or the color which are say the nitrogen here is color coded as blue, the oxygen here is color coded as oxygen. But how the computer or the system will understand this is the molecule. Okay, so there is something called as force field. Again, that is again a whole together complete lecture. So you have to actually calculate the force field. Force field is nothing but energy of the system based on its the bond length, bond angle, torsion angle, the non-bonded interactions. Okay, so you definitely need to understand. This is a way of, in layman's term, I am saying. This is a way of telling the computer the dot in that particular space is a carbon, and the dot next to that particular space, which is color-coded nitrogen, is nitrogen. Okay, so now the computer understands that this is a carbon molecule, this is a nitrogen molecule, and this is they are now connected. They are connected with single bond, double bond. All the geometry is understood. Okay, so next step is basically when you clean this particular structure is is to apply force field. Okay, so you have to click in this simulation tab and the apply force field. That will apply the force field. Now you can see this change in structure, what is happening. Earlier the structure was not without any hydrogens, now the hydrogens are being added. Okay, this is just one of the effect. ऐसे नहीं करोगे कि force field डालने से ही hydrogen आ रहा है. You can externally add hydrogens as well. This is just the net effect of the applying force field. Okay, so you get the hydrogens added. Okay, as I said, this is still not a correct geometry-wise structure, so you need to relieve this shape. You save this as a certain molecule, say mol2 format, and open this ligand in another software called as Avocado. Okay, this is again a software which has various applications. I have been using it for energy minimization. As I said, the energy minimization in this case, a bad structure. Say we have been studying this. Chemistry people will relate the pole conformation, Gauss conformation, the chair conformation. So the least energy structure is always the chair conformation, and in between you will have that. So what are you doing in that energy structure convergence? You are changing the torsional angles. So basically here also when you are doing this energy minimization, you are changing the torsion angles, the bond lengths, the bond angles, and other parameters. Okay, so as you can see this arrow, the highlighted one, using this particular button you can actually energy minimize that. You have to click on the start button, clicking on this, these options will appear. You have to click on the start button and you will see that, I'll toggle between these two slides. You can see the change in the geometry of the structure. Sorry. Change in the geometry of the structure. So you have to stop this particular when you see that the energy change is 0. Okay. Again, that is all again a different story to tell that why we have to stop at this particular point. Just for now sake of this particular workshop, you have to stop at the energy change is 0. Okay. Now we have minimized the structure. That is been done. Okay, so again you have to save this as a mol2 format, and in the same manner, okay, I will not again run through this receptor part. In the same manner, you have to go onto the this PDB site, download your receptor structure, and all those steps, that is the adding of or the isolating your part of the molecule of interest that you have to do, and save it as in apply force field, then the minimization, energy minimization, all those things you have to do. Okay, and for ligand you have to save it as mol2 format, and for receptor you have to save it as a PDB format. Okay, then we come to the actual docking. Now before we start, autolock, when we say when we go to Google and type autolock, it comes एक डाउनलोड पेज है. That is not the actual, or rather that is the actual executable which we runs the system, but that is not the actual user interface which you are using to perform the docking study. There is something other called as MGL tools. Okay, that you need to download. In the first slide, if you recall that I have mentioned that there is two things you need to download. That is, one is AutoDock and the other is the AutoDock tools, which is also renamed as MGL tools. Okay, so there people actually at the first step itself, अगर कोई download करना चाहे, that when you go to search and they stop at that particular moment. Even that happened with me when I was in my masters, that I clicked on that and nothing is happening. Okay. Then I realized, no, that is the background executable which actually runs this particular software. The main software is AutoDock tools, where it is actually the user interface. So this is the user interface which we are talking about. You have to go on file, read molecule, browse for the receptor on your system. Okay, so it will load this particular molecule. The receptor is beta saccharide. Now you have to assign the AD4 atom type. As I said, for this particular software to identify that particular dot, this particular dot, or this particular line is a single bond, a double bond, this is hydrogen, this is nitrogen. You are assigning that, or rather you are telling the software that this is the atom you have to type it as. Okay, so this is the 84 type atom type. Next, you have to save this as PDBQT. Autodoc requires the files to be in a particular format called as PDBQT. Okay, that PDBQT format, it has to be there. Next step is, now we are done with the saving it as PDBQT, that receptor. Next is with the ligand. You have to open the ligand for this. You have to press on this ligand portion. Okay, once you click on this, the several menus will appear. Then you have to press on input and click open. Again, you have to browse through your saved ligand, which you have minimized earlier. That ligand you have to select, and it will ask for this particular option. Just click OK. Okay, this is nothing but the number of polar, non-polar hydrogens, the charges which are there. It is being calculated, and that is there. Okay. Next step is the torsion tree. Okay, now you must understand how docking is performed. Docking is performed not just by placing, say this is my receptor and this is my ligand. It is not just positional thing, it is also changing the internal geometry of the molecule. So I will twist this particular torsion angle. Say again a very crude example. If a cyclohexane molecule is docked, even the chair and board conformation is docked in it. Okay, so you need to identify what are the rotatable bonds. What is happening in a cyclohexane chair and board conformation? The molecule is rotating internally. Okay, at flip. So that you need to specify that this many rotatable bonds are there. So that is what we are doing here. That set the number of torsions. Okay, by default it calculates what are the total number of torsion angles are there. That is the number 4 is there. Okay. Now this molecule you need to save as PDBQ. Okay. Till now what we have done, just a recap, we have downloaded 2 structures of receptor and ligand. We have applied force field, tell the system that this is the molecule you have, that is this is carbon, this is nitrogen, the energy calibration. The crystal structures are distorted, so we minimize that particular structure. Okay. Now Autodoc does not recognize those structures in that form, so we need to convert those structures in a form which Autodoc recognizes, that software recognizes. So what we are doing is saving it as a format in which the Autodoc can recognize. So both the molecules, the receptor as well as the ligand, we have saved it in a PDBQT. Right? Okay, this is where the actual docking starts. Is using first is the grid calculation. Okay, we'll come to this. What is grid? When you understand, I will show the grid box. So sorry. So you have to press grid, then select macromolecule and open. Okay. Now whatever the PDB molecule you have, a PDBQT molecule you have saved, that you have to open now. Okay, so when it opens, it comes with this pop-up again. You have to click no here. Okay, click no here because it is asking basically the charges and the atom types which are to be calculated. So you have to click no here. This is again a warning. This is not a warning as such. Now, mind you, in any computational technique or any computational method you are doing, warnings are okay, errors are not okay. Okay. So if you get error, you have done some mistake. Warnings are still okay because this warning is coming because of the compatibility issues. A certain molecule is compatible with certain software which is not compatible with other software. So warnings are okay, errors are not okay. Okay, now you have to define what is your ligand. Now you are actually in the docking protocol. Okay, first you have defined this is my receptor, now you are defining this is my ligand. So again you have to open the ligand, and next the important component of docking, where you want to dock your molecule. Okay, say again this is my receptor, this is the region which I want to dock, this is the region which I want to dock, or this is the region I want to dock. Okay, or I want to dock the entire surface. As in this case, I will mean that. So you need to specify what is your binding cavity. That is what we are doing here in by specifying the grid box. Okay, so you can see the beta cyclodextrin is surrounded by a box. Okay, which will tell the system you have to dock in this particular area only. Okay. Now, when you are looking at, say, certain large molecules, say a protein receptor or enzyme, which are very macromolecule type of, you putting a grid box on the entire molecule would be computationally expensive. Okay, when I say computationally expensive means it would take many more days or much more time to be invested. So basically we depict this in a particular concise one. But now, since this is a very small molecule compared to the proteins, we have defined this as the entire beta cyclodextrin. So what will the molecule software do is take your ligand, that is your ibuprofen, and dock to the entire surface. Okay. Now it can see, if you see closely, that it is defined over the entire surface. That means the molecule can stick outside of the beta cyclodextrin as well as the inside, inner core. Okay. So you are not sure whether this is happening or how it will get out. We are coming to the end. Sorry if I am lagging because there are many elaborate steps are there. So in the next you are the next step is to define the output. It is just a parameter file which is used to calculate the grid. So here, this is where the mistake is done. That when you are specifying in Autodoc, when you are saving any file, you have to specify the extension name in when you are writing the name of the molecule. Say I am saying beta cyclodextrin dot GPF. That dot GPF you have to type explicitly. Otherwise, just word save करते what we do, just we save it as. You don't look after that. You don't write it as dot doc or dot docx. But for this particular software, you have to do it. Okay. One more thing, if you are not aware, because this is a Linux environment. Okay, this software was first developed for Linux environment and then it was migrated to Windows. You have to make sure that all your folders or any molecule which was specifying the name does not have a gap. Okay, say the folder name which I have mentioned is ATAL FDP. How I have written is ATAL underscore FDP. I have not written ATAL slash or space FDP. Even when you are specifying ligand 1, okay, ligand 1, ligand 2, ligand 3 or receptor 1, 2, 3, there should be no spaces. Okay, उसमें ऐसा है कि Autodesk cannot find the file. Okay, you will find that particular that because it has spaces, it will not recognize. So that care you should be taking. The naming of the molecule, naming of the folders without spaces. So you have to click save here. Okay, so that is done. Now we have to run the auto grid. Okay, now in a brief manner, very concise manner, I will tell you what it is doing in this case when you are running the auto grid. Okay, now the box which you are seeing, it is a hypothetical box which is placed over the molecule, and you are also specifying that this is the receptor. So for every atom type, when I say atom type, carbon, nitrogen, oxygen, whatever the atoms are there, the interaction with the hypothetical probe and this molecule is calculated. Okay, so that calculations are used in the docking purpose. Next step is to run this. Okay, now again, this particular window which you are seeing is also very important. Okay, you can see that the file location is D administration, that is my D drive administration ATAL FDP. Okay, and all my files. Okay, this is again a mistake which the researchers performed, that when I install, by default that installation, the Autodoc or AutoGrid exe, AutoGrid exe goes in a C folder. Whenever you install any application, by default it goes in the C drive. Okay, here what it does is you have to specify when you are using Autodoc, all your files should be in one folder. Your Autodoc exe, autogrid.exe, all your ligand file, input file, that should be in one folder. Okay. The software get confused कि एक फाइल इधर है, एक फाइल उधर है, it cannot match. So basically what you do is pull all your files in one folder. Okay, so you can see that all these my files are in the same folder, FDP folder, my grid parameter file. So when I browse for this file, this glg which you are seeing gets directly automatically generated. Okay, so that you need to take care that. So when you click launch, it performs the calculation at the background. Okay, so it background. Okay. Now you have to search for in your folder. Say ATAL FDP is my folder. You have to search for this glg file. Okay. Now this is again important. You need to go open it using a word processor or word pad and go to the end of the document. You should see this message: successful completion. Okay. If there is unsuccessful completion, that means there is some error. So unless this successful completion is there, the auto grid calculation. Okay, now what I have done is I have specified this is my ligand, this is my receptor. I have done certain grid box calculation, or rather told this system that this is the place you have to dock. Okay, and these are the energetics you have to use. Now the actual docking will start. So edit, you need to delete all molecules, although you can do in the same step, but I will prefer that you delete all molecules and start over again. Next, you need to go to the docking folder or the docking tab. Here again, you are specifying what is your macromolecule. Okay, so that you have to specify by choosing the macromolecule which you have specified. Okay. Next is you have to choose the ligand, which ligand is to be taken. That is your drug in this case. That is being shown here. Okay. Now you just have to click accept. These are the certain properties which are calculated, what are the atom types and all those things. Okay. Now search parameter. Search parameter, there are many parameters. This is a heart of the docking. What search parameter you employ will alter your result. Okay. Though there are many algorithms which are not going to detail. We are looking at a genetic algorithm. Okay. So basically what it does is the number of G8 runs is 10. Okay, so 10 times it will search for where it is actually fitting. That ligand and the receptor where it is fitting, it will search 10 times. You can change the number of this particular position or this particular. So depending on the number you specify, the time will increase exponentially. Okay. So you have to click accept. The default values also you can use. Okay. Then you have to specify the output file as landmark and GA and save it as again. You have to explicitly type .dpf that care should. And then you run the Autodoc again. Care should be taken that all your files are in one folder when you are looking at this. Again, whether that once that launch system is there, a window will come. Okay, sorry, I could not include that. A window will come and it will at the bottom of the window, dismiss option is there. Don't press that dismiss option. That will kill the job. Okay, in computational we use that kill the job. Actually, this is the word which we generally use. Kill the job. Okay, that means it will stop the docking process. So don't press that dismiss. Okay, let it happen. Okay, how do you come to that that docking is completed? Okay, so there is a file called as DAG file generated. That DAG file you can see is here. Again, you have to open that DAG file and you have to search for go to the end of this file and you will find that successful completion message you should see. And what are the results of this? How do you get the results of this? Is, as I said, the software, because you specify the value of 10, it will search 10 times. Okay, It will try to fit in the ligand, or in this case ibuprofen, into the cyclodextrin, SA, all those different poses, and it will calculate the binding energy. Okay, this is again an estimated binding energy. So, and it ranks this in terms of this particular, so you can see the ranking. So from this you can see that the first position, now you should understand this is binding energy. More negative is better binding. That means numerically more negative, -10 and -7. -10 is more effective binding, -7 is not more effective binding. So this is what we get. Now, our actual aim or objective, we started with whether ibuprofen will fit into the beta cyclodextrin and in what ratio can be fitted. Okay, so this how you can do is result analysis. Is there is a question called as analyze tab is there. So using this particular options, analyze, walking, open, you have to open the DAG file which you have created. Browse for DAG file. It will open the confirmations. Okay. Now you can analyze your solutions in this particular software, but it is very tedious. It is not publication friendly. So what I do is I save this as a file, PDF file. Okay, and open this in the Discovery Studio Visualizer. Mind you, you have to open both your ligand as well as receptor file, that is cyclodextrin, and the ibuprofen in the same window. Okay, now you can see how it has docked. It has docked very nicely. That the cyclodextrin has in the center of the molecule. Okay, this is just the another variation of the pose. Okay, cross transfer section, you can say, that is all other longitudinal section it is being shown. Okay, so this is how it can perform. Now if you want to check whether drug, two molecules and one cyclodextrin, what you can do, treat this complex as your receptor and again dock your ligand, ibuprofen. Okay, if that is fitting, then definitely it is forming 2:1 complex. Or if you want to check another molecule of cyclodextrin can fit into this particular complex, you have to take cyclodextrin as your ligand, treat again this complex as your receptor. Okay, with this, I'm sorry if I've taken too much of your time, but this is I wanted to take, thank you for your patient listening. Okay, and these are the computational services offered at Desktop. You can definitely collaborate with us. Molecular docking, QSR study, molecular dynamics simulation, P energy calculation, and rate energy calculation. Thank you all.
Thank you so much, Dr. Mushtaque sir and Dr. Anand sir. It is really a nice session. I need to have a class with you after this. Definitely, really good session. I got a lot of information. Thanks to all participants. With this, we end for the session of the day. Tomorrow, all should gather at 9:15 for the breakfast on 6th floor and 9:45 for session. Okay, thank you.